Thank you for the invitation to present. I'm happy to see here in this community and this audience some very good and old friends that who have worked or interacted intellectually in the past. So I'm going to discuss with you parts of our work on methods and resources for sustainable bioindustry development. So this is work, of course, this is one snapshot from the group. This is one of the best pictures we have of the group with a nice view. So, of course, the principal funding sources are from the Swiss National Foundation and from European grants. So we have, over the years, we last, I would say, more than 20 years, we have a big academic network, European and international abroad, and also I've been working a lot in different types of forms with broad spectrum of industry that ranges from the energy to nutrition to health. So I'm going to discuss with you about some aspects of our works on the three pillars of sustainable bioindustry development. Of course, there are more pillars, but from our perspective, there are kind of three pillars when we look from the work of the analysis, computational modeling analysis of metabolism, which is the pathway discovery and reconstruction, pathway design implementation, and then whole cell biocatalyst optimization. So I'll give you some vignettes from some of our work in here, some of the, of course, foundational work, and also some of the highlights of some interesting cases. So the pathway discovery. So as we all know, this is the common biochemistry image that most of us, we grew up with posters like this in the labs. So and when the internet came, everything was organized into bioinformatic tools, and like the metabolic maps, for example, in KEGG, which again organized in pathways, and within the pathways we have different enzymes and reactions. So of course, as you're all very well aware, there is a big domain of applications from agriculture to metabolic engineering to drug design and toxicology. So as we venture now into these uncharted territories, can we distill the spirit of biochemistry and navigate this metabolic complexity? Towards this, of course, we're a computational group. We say, can we go from the knowledge we have in reactions, especially on the type of chemistries we have in reactions, from in vivo to in silico? And we say it would be nice to have something like this. And then we develop a method we call bnice.ch. As I like to say, that doesn't matter what the acronym stands for, as long as it sounds good. These are work that over years we've been developing since some preliminary ideas even that I had working in Cargill and DuPont. And I estimate once that it's more than 100 FTE years that have been developed in this work. So the whole idea there started when we realized when these pathway kegs, the pathway maps, they came in keg, we're looking for these reactions, trying to guess which enzymes, sitting together with organic chemists and enzymologists, which enzymes might be able to do some to act on new substrates, to make some man-made chemicals, to essentially that was from the very early days of sustainability, before the name even was existing. So and then we observed that there are the key observation there is that if you look in the enzyme commission classification system we're all familiar with, there are these 4-digit numbers. So the first digit is the general type of chemistry, like oxidoreductases, transferase, hydrolase, lyase. Of course, now there is a seventh one, which is for the transporters, but it's not really kind of reactions. But for us, it's mainly biotransformations that we're focusing here. So the second level has to do with what functional group is acted on this reaction. For example, the 1.1 is an oxidoreductase that acts on the hydroxy group, whereas 1.3 is an oxidoreductase that acts on a carbon-carbon group. The third level, again, is still general and denotes has to do something with the cofactors or co-substrates that are present in these reactions, okay? For example, 1.1.1 is an oxidoreductase that acts on hydroxy groups using NAD or NDP as acceptor, whereas the 1.1.3 uses oxygen as acceptor. And then the fourth is specific essentially to the substrates that we have in this particular reactions. This is now specific to the enzyme and not anymore on the chemistry of the particular reaction that we have there. Now we'll say, okay, so if we look then at closer at the databases, and then we see in those reactions that essentially it's the enzyme classification of the reactions. Then we see, for example, that reactions like belong to the same third class, 2.7.1, they actually do act on different substrates, but in reality, the chemistry is happening on a small subsection of the molecule, which we call the reactive site of the molecule. So this led us to the formulation of the generalized enzyme reaction, which essentially the generalized enzyme reaction considers not just the whole substrate, but only the reactive site that is specific to the third level. And in this way, we developed this method, biochemical network integrate computational explorer method, and the .ch stands for Swiss quality. Of course, the idea there is that we go from in vitro to in vivo, we know the metabolites, we know the enzymes, we get the biochemical reaction. The question is, how can we encode this reaction, and then how can we translate mathematically this enzyme, generalized enzyme rules, such that upon the action of these rules, we can start. So we have encoded now these rules. So we went down all the known biochemistry, and we extracted 450 generalized enzyme reactions that represent almost all the known biochemistry, 99% of the known biochemistry. And of course, it's ×2 because it's the forward and the reverse. And then the idea is that now we have a database of reaction rules. We encode the molecules, we encode these rules, and then we throw in a chemical. So upon we throw the chemical, then the algorithm, the program says, the BNICE, uses these rules and it checks every possible site on the molecule, and it says, I have this particular group here, then I can implement the generalized enzyme reaction rule 1.14.1. And on the way, we also produce the chemical. So essentially, it is the mechanism without going into how electrons move around, but essentially it's the input-output bond breaking and bond making. So, and then searches more, and turns out that even in the same subsite, there is even a smaller subsite where the 2.1.1 reaction can act. And this way, essentially, we can go screen all the molecules we throw in, and then we find out what possible reactions are, what possible enzyme reactions. And then this is the first generation, which means that we call the first pass. The second generation now, we take the product molecules of the first generation, and we again screen them, and we apply this rule. So in this way, what we're able to do, we start with a set of molecules, we start with a set of rules, and you can use all the rules we have, or you can use a subset defined rules. And then you produce everything possible that can be produced if you had this IJK element QRS rules acting on A plus B. And then in the second generation, you apply again the rules, and now you produce, again, of course, the same molecules, but also you produce the novel molecules, which are themselves the products of these enzyme reaction rules on the products of the first generation. And this way you can build these rules. Actually, what is interesting here, we can make the following observation. Then we can say in the rules, in the molecules that we produce, we can classify the molecules in 3 classes. One class is essentially that we start with some biochemical, known biochemicals, and then we produce known biochemicals. However, here is that we have 2 cases. Either we produce the known reaction, which we know we reproduce because we have, this is how we built our rules, but also we produce novel reactions that have not been reported before. And now the products, they can be classified in 2. They are themselves known biochemicals, or there are chemicals in the chemical databases, or they're what we call BNICE compounds, which are completely new. So ideally, somebody would like first to investigate compounds that are known rather than novel compounds, but of course this is not the limit. So we say, how can we create now something that can be really useful, that somebody can use to mine without having to deal with running all this kind of rules? So essentially any kind of user can come and say, okay, I want to go from A to B in the known biochemistry. How can I do it? So towards this, we created the Atlas of Biochemistry. The Atlas of Biochemistry, what we did, we took KEGG, a database, as a reference, just for benchmarking the method, developing the method. And we took all the rules. And then we say, if I apply now my rules in every possible chemical that exists in a KEGG database, in a known biochemical database, and I keep only products that exist in the same database, how many reactions? So essentially, without increasing the metabolic space, without increasing the chemical space, we say how many new reactions we can have. But be careful here, these are enzyme reactions, so reactions for which we have even an easy number. And this is how the KEGG looked like. So we have 16,000 compounds. Many of them are disconnected. This is very important. These just have been deposited in database, have been identified in a biological specimen. And then, of course, we have also the and then we have also sums that are connected with 10,000 reactions. So now when we apply all the reaction rules, then of course we created a much denser network. We were able to connect orphan compounds. So essentially now we can even postulate what type of reactions have created these compounds and what were the starting molecules. And as you can see, we have increased the reaction space by 1 order of magnitude. Now we can all understand here, of course, we did the test. We say, okay, how do you do this test? We took the KEGG database of 2014 and we compared it, and we say, can we predict the new entries that we're going to have in the database of 2019? And then sure enough, we were able to predict 66 of the novel reactions that came in a later version of KEGG. This is typical bioinformatic kind of analysis. So now somebody can go there and essentially can ask the question, well, how many ways I can go from A to B, known biochemical compounds, but now not only with the known reactions, but also with novel reactions. And of course, we would like to expand the concept, and towards this, we created the Atlas X. The Atlas X now, what we did, we include all the available and well-curated, that's also important, compound biochemical databases, which are, they have a total of 1.5 million compounds. So these are compounds that exist, distinct compounds that exist in these databases. And then, of course, in these databases, you have the reactions, and that was really a Herculean effort because as you can imagine, within these databases, we have a lot of common entries. And as you know, those that work with databases, that every database has their own identifiers. So it has been a big effort. But the nice thing now is that we even have a database that we have all these compounds that somebody can go look at them. And we have already a mapping to the identifiers in every possible database. So essentially now you see an amazing resource, even at this stage, for somebody to look at as a database. And now we start with KEGG. Remember, we had 140,000 reactions starting and only the 16,000 compounds. But now we increase the compounds because we have all the chemical and what we call the bioatlas, which are essentially all the biochemical known database. And of course, we included not only what has been reported themselves to be compounds that are this includes not only compounds that have been reported in biochemical reactions, but also bioactive compounds for obvious reasons, because we want to understand their metabolism. And then also we allow the reactions to also produce chemical database. So the next step now that we'd like to do is now to apply these methods to PubChem. And it would be nice to ask the question, okay, given all the chemical space I have, how many enzyme type reactions I can have application to them. So, and then of course now you can clearly see on how you can use this in order to identify synthesis routes for man-made chemicals. And I'll just give you an application, and I like to to show stuff from our collaboration, but I prefer here to show you that people used it outside our collaboration as a resource, and they looked in the conversion of C1 compounds into biomass building blocks or compounds, and of course the systems they were using, they were very they were lost in arrival because they were already losing some of the carbon in the early reactions as they were going to assimilation. And then they say, can we avoid this CO2 or carbon loss, and we can get most of the yield out of it, and more, of course, of energy in the system. So they looked at possible reactions that could possibly do that from known databases. And then they also found that we had in Atlas predicted 73 novel aldolases, and indeed they integrated these, and they even engineered these new steps in order to do it. So you can see here a demonstration that people committed, they asked the right question, and they went all the way to engineer enzymes that will be able to deliver these new reactions. So the second step then now is, once we have this database, okay, how can we use now this database to do something more? So one of the things is to discover novel reactions, novel enzymes, solve particular problems. But can we use it for pathway design? So I'll just give you some of our recent work that it has been done within the effort of a European Grand Shiki Factory, where the project, the objective of the project was to find routes to produce 100 chemicals. And the starting kind of hub, it should be around Shiki made. So as you can imagine, a lot of aromatics, a lot of difficult. So some of them were biochemicals, but not known to be connected with reactions. Some of them were chemicals. So, and then our method was able to indeed, was the only method within the consortium that was able to find for all 100 chemicals to find routes for this. But now, once we have this database, which has been published as a special database, Arbor metabolic engineering. So now we have this reaction network. This is the Atlas type of reaction. So what we do is we say, okay, I want to go from phenylalanine to benzylcinnamate. Okay, that's one of the target compounds. So what you can do is that you can do a linear core pathway search. Now, when you do the linear core pathway search and you try to put this inside your organism, you have to be very careful that, of course, you're going to have some novel compounds, but you have to make sure that any kind of side cofactor and co-substrate, it is also exists in the native metabolism. Because, okay, you can introduce this reaction and this reaction to make this, but if your input is not native, then you also have to identify, can I have also, which additional reactions I have to add? Because many of the times when people apparently they try to do this, they are missing because the host itself does not possess this particular compound that needs as a site. And here is, so what we say we developed this algorithm, which we call SubNetX, where essentially now we use not only to extract from these databases the linear structure that can take us from A to B, but it can extract the subnetwork, which once embedded into the host, all the inputs that we can have into this host is going to come from the particular compound. For example, here, one will say phenylalanine, okay, I can make to scopolamine. But now you can see at certain point I need tropin. Okay, so essentially, is tropin present? No, tropin was not present in École. So this means then we ask the question, can I use again my database to find another network that it connect me from tropin to something that exists in École, and on the way, I don't have any cofactor or substrate to do that. And then once you have this, now you can embed it in these known genome scale metabolic models. You can embed it in your yeast, in your École, and then you can say, okay, I can guarantee that at least once I put in my subnetwork, this will be functional because the organism has all the boundaries, so to speak, metabolites in order to carry this reaction. And then you can do this analysis. You can study what is the size of the different networks and the yield that you have here. And as you probably know, our group has pioneered the use of thermodynamics in biochemical networks. And when you have a hammer, everything looks like a nail. So for everything we do, we put thermodynamics. And you can see also here very nicely how when you put thermodynamics, you definitely, as you would expect, you get different types of networks. For example, you need longer pathways to make the same yield, whereas without thermodynamics, because you violate some, so to speak, of the energy constraints, you get shorter pathways. Another interesting thing, and we can discuss later if you want, but I find very interesting, you can see here a lot of these noble compounds that we have, the number of the reactions that we need to add in order to improve the yield, it is really increasing. So this is very important now, because imagine now the metabolic engineering strategies, the interplay you have between the effort you have to do to engineer the pathway versus the return on the yield. And this, of course, this kind of increase is not straightforward. Usually you are thinking that shorter pathways can give you higher yield. But in this particular case, because we have to, in most of the systems, to engineer additional subnetwork reactions that will support, let's say, the production of the target chemical from the or host organism, then this is caused this kind of linear, this kind of dependency. And then, of course, we have a method, Bridget. I'm not going to discuss about it. Okay, I have some slides for later, but further we developed now a method which is very successful that help us now to identify sequences for this novel reaction. Because when we have a hypothetical reaction using the be nice method, we don't have, because it's a hypothetical reaction, we don't have a sequence for this. So we ask the question, is there a method that we can use in order to be able to identify this particular sequence? And we can discuss later if you want, but it has been really very successful. So then we move to the third pillar, whole cell biocatalyst optimization. So, and I present here kind of the people that work there. Senior scientist at our lab, Ljubis Samiskovic, has been instrumental in the development of this work, and a lot of pioneers like Li Qing and Alexandros that have really worked in development that led to the results I'm going to show you. So this is a typical case that we have in whole biocatalyst development, metabolic engineering, synthetic biology, this kind of approach. So the question is, we have a reference state, we have a certain specific productivity from an organism, and we want to improve the productivity. What is the target? How can we find the target? In our case, of course, coming from the modeling and computation aspect, we say, given a stoichiometric model and the reference physiology, a flux profile, a metabolite profile, partial information, how can I go to a target physiology? So the core idea is, we're all familiar in this field, is identifying what is the rate limiting step, which means essentially which is the enzyme that we should change to increase the pathway throughput. Now, if it's a linear pathway, it's very simple. Without any feedback control, the rate limiting step is the first step, because no matter how much I change the enzymes 2, 3, and 4, there's no way to push more if I don't put more. Now, as we go then to more complex stoichiometers, even in this case a branch pathway, then again, it's not very straightforward. So therefore, we need to know, even for this branch pathway, not to identify which is the most rate limiting step for the production for E or for L, then we need to know kinetics. But the problem is that the kinetics, a lot of people say kinetics, then we should measure everything in order to estimate the parameters, because fortunately it might exist a lot of databases out there that they have some kinetic information, but the problem is that that essentially is limited, is specific to organisms, is very limited to specific subsystems. Even for whole pathways, it's not for every possible enzyme. And if you really go close and look at this data, you might have the same enzyme, but the biochemical method, the assay developed, it is not the same. And it's a little bit embarrassing when I see a lot of people using this data to do any machine learning training when they have not calibrated first the temperature, the ionic strength, the conditions where these things were measured. So essentially, we do a lot of estimation right now using all these machine learning methods, using the right data, but under the wrong conditions. That's very critical, I think. So even in this particular case, you can see that we have 20 parameters just for this simple pathway. So essentially, we need at least 10 independent experiments, dynamics, steady states, in order to estimate it. So the reality, we're faced with partial measurements, number of fluxes. We can never measure always all the fluxes, or we don't have the resources to do it. The number of metabolites is going down as we go, the size of the system, and of course the number of the experiments. So the system is terribly underdetermined. So in this way, we propose that instead of trying to find the best kinetic model for a partial set of data, is to find what are the possible alternative models that essentially they can be consistent with the partial data. Because every time you have partial data, then this means that you can have many alternative models that can explain the same data. So how do we do this? If you look in the field, not only in our field, especially in computational chemistry and engineering, more than 100 years ago, when people start using computers, they start using a lot the sampling method. So what you do, when you have missing data, you start generating data, try to fill in the gaps by some data, and the way you generate data is with some certain constraints that obey some certain physiology. So in this particular case, also, so we want to build a kind of kinetic model for this particular system. And let's say the only information we have here is a simple exometabolomic information. How much is the uptake and how much is the production rate? And here, even in the bottom, I have made even a mass balance here. If I measure only the production of one of the compounds, how much is the other? So now we're facing with these problems. We have the complexity, the size, and nonlinearities, and then we have this uncertainty. Towards this, we've been developing since 20 years now a workflow which we'll call Oracle, optimization and risk analysis of complex living systems. What essentially does is building population of kinetic models that are consistent with partial information. Because the idea there is that if I have these partial models, then I can do at least a statistical analysis. I cannot know exactly what is my rate limiting step, but I can say what is the most probable rate limiting step. So essentially, the trade-off for model uncertainty is the multiplicity we have, and still, I think if we're doing good job, I think these distributions we get are very tight. So essentially, we get the fluxomics. Now we now have partially known kinetics. This is the enzyme activity. This we'll never really know because it's not only even if we know the K-cut, we never know the total enzyme that we have in the system, assuming the simple kinetics. Now this is a thermodynamic displacement term, and this is the enzyme saturation. And this is the simplest uni, one reactant, one product, reversible Michaelis-Menten kinetics. So now what we do, so from reaction thermodynamics, we can estimate the K equilibrium. Maybe from metabolomics, we can estimate some of the concentrations, or we can do sampling. But again, if we do sampling, it's not we can do sampling completely out in the space. For example, we might know either from experimental information or from hypothesis or from prior knowledge, from expert knowledge, for example, that the concentration of the A, B, C, the intracellular concentration, it is bounded between certain concentrations. But the thing is that we also know that the reactions have certain directionality. This means that the reaction runs from S to A. Immediately, the reaction directionality coupled with the delta G of the reaction tells us that the substrate cannot, the concentration of A cannot be higher than a certain concentration. Otherwise, the reaction would have not been able to run in the right direction. So using this thermodynamic constraints that we have in the system, with the methods that we have developed over the years, now what we're able to do is, even if we have a big space of hypothetical ranges of concentration, we can identify the subs area where it's the thermodynamically feasible concentration. This reduces immediately the size of the metabolite space. And then if you have partial observation for, let's say, the range of one of the metabolites, you can even cut low. So essentially, then is that all the kinetic models we have to build, they should be consistent with this concentration. So what we do now, we have a known maximum activity and a known saturation term. And then this one is something that we can get from the thermodynamics and the sampling. So, but here again, you can observe that this saturation term in every possible kinetic of the enzymes, because this is the heterogeneous catalysis enzymes, you always have a saturation term. So this is between 0 and 1. So essentially, instead of sampling on the unknown parameter space, we do the following. We sample in the saturation space between 0 and 1. We sample the metabolomics, and then we back calculate the binding constant. So in this way, we can calculate the saturation term, and from there we can calculate the Vmax. So that's how it works. We take this, we sample the concentration space, we calculate the ranges of Ks and Vmax, and then we get essentially distributions like this. And this gives us all the possible combinations of kinetic parameters that can explain this particular set of exametabolomic data. Now you can appreciate this, that at least we were able to put some kinetics in the models. Okay, but of course we would like to have models that are meaningful to serve our objectives without ad hoc assumptions, without violating principles, without loss of information, and the ability to build these models in the different sizes and scales, and of course to have a method that would be reproducible across lab. Because a lot of the times when you see kinetic models out there, it is really every lab has its own kind of way. So there is not a standard operating procedure. Therefore, we propose this workflow, which we call the Oracle, where we start with the stoichiometry, thermodynamics, some metabolomic data, some fluxomic data, and then we start doing the thermodynamic analysis to sample the concentration space for those that we don't know, and then we sample the kinetic parameters, and then we build population of kinetic models, and then we use experimental observations or expert knowledge to prune the data, because you know some models, they will behave, although they will fit your exametabolomic profile. If you make them a dynamic simulation, they don't fit the time scale of your system. And this way, then you can make metabolic control analysis to find the rate limiting steps in this network, and then you can design alternative engineering strategies. Now, the valid kinetic models should be consistent with observed physiology. So if you ever get anybody gives you any model that is not consistent with the physiology they have, this is very important. One of the other things that we find ourselves is very important, a lot of the models that you see out there, they are not locally stable, which means that if you make a slight perturbation in the concentration, then you get completely in a different steady state, you get completely different profile, so your model is not robust. So also, the other thing which is very, very important, and this is known since the early times that people do, since the Heinrich and Rappaport days that people were doing dynamic models of metabolism, and Shalkov and Reich, that essentially metabolism has to have a certain time scale. And what we call here a biological time scale should be much smaller than the doubling time. Why? Because the thing is that when we observe experimentally the system, when we even report metabolomics, we assume that the system operates in a quasi-steady state, in a pseudo-steady state, which means that when I have perturbations in the Vmaxes, in the parameters due to division, etc., my system very fast, in a millisecond, essentially converges back to the steady state. Now, if you take a lot of the models that are consistent with stability, are consistent with fluxomics, what you find out that some of them, they have very sluggish response. So you know these models don't have physiological kinetics. So this way, we can see that we can put all these arguments of the quality of the models, and then we can start building these models. So we start by the usual kind of Oracle that generate all these models, And then we do this kind of screening based on the stability, on the nonlinear responses about the time scales, and on the different robustness. And then we go and do the strain design, because now we take these models, we immediately reduce the number of these models, but now we can go and do now identify rate limits and step to do metabolic control analysis in every model, and we can identify the response of the system to small and large changes, because we have a full nonlinear representation of our system, and we know our system is robust. So even if we make large changes, the system will come to a reasonable steady state. So this overall is the outcome of the oracle, was essentially we have population of kinetic models consistent with thermodynamics. Thermodynamic displacement of the reactions is preserved, and also they are better characterized with respect to the fluxes. And now in this way, essentially, we have also now this is the timeline that we started the last 20 more years, that all this development, and now we have models, genome scale models. We say genome scale, we can go up to 5,000 reactions, but always we try to keep it on reactions that are relevant, and we can discuss this. So this is one case. We have also models for yeast, which is used for different projects. We have for Putida, and even we have for human metabolism, which is now the human metabolism is in the range of 1,500 reactions. And we have used these models. In the case with Genomatica, we used the École model in order to identify rate limiting step in the production of 1,4-butanediol. And from what we hear, it gave some very useful suggestions for strain development. Of course, we have worked with Meria in the past in BDT, and we investigated the type of manipulations we have to do in order to optimize the biomass utilization, xylose and glucose towards ethanol. And with DTU Biosustain, very recently we published this paper in P-cumaric production using yeast, again using the whole yeast model to identify successfully targets to manipulate, and also worked with partners to using Putida. What I want to do with you, I'm not going to go read the papers. You can get also the coupling with the experiment, but I want to tell you about the method that we have developed, which is very critical, and you will see why. So this is what we call constraint-based metabolic control analysis. And this is actually motivated by, it's based 95% on the work of my thesis, where my co-advisor was actually also Chris Frudas, who was Antonis's PhD advisor. Of course, I'm younger than Antonis. And then what you can see here, in the paper say that the concentrations of metabolites, Jay was very particular about, because if the early days of metabolic engineering, they were shooting their feet by just overexpressing, with any kind of promoter enzyme. And then what they had is that the system will go completely crazy, because they should neither exceed toxicity levels low. You should preserve cellular activities that are required for growth. And therefore, these all responses of the system should be constrained. So essentially what we did here, we translated the method we developed in the mid-90s. We translated now to a formalism that is now be able people to use easier using MATLAB and Python, that they can essentially do what we call network response analysis, NRA, which now it says that, can I maximize a particular objective subject to physiology constraints and concentration constraints? For example, given an objective, I want to optimize tyrosine, which four or any number of enzymes I should manipulate up or down without compromising the cellular, without losing growth, maintenance energy, homeostasis. And here we have a work that we did based on some data, public data, where we apply this method, and we start with generated 1 million almost. I mean, we could generate 1 million, but let's say 800,000 models. And then based on the dynamics and stability, they went down by 1 order of magnitude. And then based on the fermentation consistency, another order of magnitude. And then we end up with 10 models. This is also what I'm telling you, that essentially we have a lot of pruning in the models. And then we have these models that we see how consistent they are. These simulations are essentially, this cloud you see is the simulations, the statistics of the simulations we get if we perform the same simulation for all alternative 10 models. And then we can do this. But then we say, which are the top 3 enzymes that I have to manipulate in order to increase my translate production? If we use just the MCA, the traditional metabolic control analysis, which means that I identify rate limiting step, and then I change this based on how good rate limitation they have without constraining the rest of the system, look what I have. I increase anthraline rate, but I get 0 growth. This is, people know by practice and also expected. But now, if we use the NRA and we say which 3 steps I should manipulate such that I can increase my anthraline rate, but at the same time, my growth, my fluxes to growth should remain within physiological constraints, boom, we get here that first we can do as many fold changes as you want, but you can clearly see that we preserve the growth that we have in the system, and at the same time we get very good anthraline rate. And you can see also something interesting. The best anthraline rate is when I have some compromises on growth, but we still allow to have it. It's not that we say just fix the growth. This is a part of the computation here. So this is, I think, for me, this is exciting results, because the thing is that none of these steps that you would have identified with common modeling and metabolic control analysis would be the same. Why? Because these guys, they also have a response, the system responds also in ways that is not physiological. You don't want to respond. And now you can do, again, with the methods we have, you can find all alternative strategies. So we have 41 alternative strategies, and then you can do data mining in these alternative strategies, and we can see that there are 5 designs. For example, I'll focus on the 4. You can see here, it tells us that we should manipulate 2 enzymes in the shikimate pathway, but then any possible combination of the enzymes here. And the same, of course, is here. So this way you can do a lot of data mining to see how the interplay between pathways and sets of enzymes evolves. So the question is, can we do better than this? The question is, can we use now to find these regions? Because as I said, we're going from 1 million models to 10 models, okay? So essentially, we need methods that can help us design better sampling to do always to find, can we find parameters to enrich our parameter space with high quality models. So essentially, we started this work even in 2016, 10 years ago, where we start by classification. So we'll take our samples, we'll classify, and by looking at this classification, we will find out which are the parameters that were essentially the most important that will preserve the performance of the system. And then you are using now this kind of area. Once we identify the area, then we do more dense sampling in the area that we know is more consistent. And with this dense sampling, we get better statistics. And of course, because we live in the AI area, we said, are there specific methods out there that can be tailored for our method? And MISCO and Subcam in the lab, they looked out there and they proposed the GANs, which is used for images, but the most important thing is how it works here. So we have the method Rekindle and Renaissance. We always have to give nice names, where essentially we generate these models, as I said, based on the criteria of stability, of dynamics, of characteristic time response, of the fermentation pruning. We identify which parameter areas are the ones that have this characteristic, but we have very few points. So essentially, we use these few points we have here, and then we train machine learning so that now we can do much denser when we do sampling to always have, now we can have up to 500,000 models that will be consistent with all the strict requirements we have about dynamics and the quality of performance. I think this is really transformational because now we can really accelerate everything, and at the same time we can move parameter space. Plus, we can now look at the distributions of the parameters. We can do a lot of meta-analysis, try to understand why these things happen, So because a lot of these things is not interpretable, so I think the challenge now, how can we turn now this kind of classification that we have from the machine learning into something that we can formulate as explanatory hypothesis for the response of the system that can help now the scientist, the metabolic engineer, the process engineer to think now out of the box and for the next. And these are the two papers that I mentioned for the methods. So now we can. The other nice thing is this turns out that this particular systems, a lot of the information is in the structure of the metabolic network or similar structure. So essentially, we can take now any kind of similar kind of central carbon that from other organisms, and it needs much less data to train the system. So we have transfer learning. Somehow the system in the structure, it keeps the information. So a lot of the things, the parameters that we have, the kinetics that we use for the system, they can go and use. So we had published this nice paper, a little bit early thoughts. If I was writing it again, I would write it differently, but it was really nice that it was demonstrated for the first time. But now we really have kinetic models of genome scale. We can go up to thousands of reactions without sacrificing stoichiometric thermodynamic physiological constraint, without doing any modeling crimes as commonly done. And actually, we can do it now for any organism in a couple of months. Overall, I think our tools, our workflows, they allow us to cut by tenfold the time and effort where we have been and where the field, some of the field is still is today. So, and of course, a lot of these methods now are used for the analysis design of microbial communities. We have a big effort here in Switzerland for microbial communities. I think the field, as Doug Cameron had predicted, the metabolic engineering is moving in metabolic engineering using communities. We work with Merck and Roche for the optimization of cell culture for biologic CHO cells and other very complex cells. And also we're working using exactly the same methods I present here in order to do drug target identification in cardiovascular and metabolic diseases. And with this, I would like to thank you for your attention, and I will be happy to answer any question.